Hey, easy, 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 easy. And he has to settle down. Good morning, Zoology 2700. Happy Monday. I hope where you are, it's as sunny as it is where I am. It's a sunny, cool day. I think the thermometer said it was minus 11 here in Guelph this morning, which is, I think, lovely. And it makes it easy for us to make ice on our backyard rink, which we like to do here. I hope you had a good weekend, and I hope that all of the things that you were going to do to take care of your mental health you were able to do. And you rejoin us rejuvenated and ready to go. There's a bunch of people in the waiting room. Hello and good morning. So what we're going to do today is finish talking about the jellies, the Cnidaria, and then we're going to do a bit of a digression into some of the assignments or some of the marking that'll be done, because there's a new assignment this year about species names that'll be released on CourseLink this afternoon. So I want to talk about why it is we're talking about species names and the process of naming. Maybe for you, this is the first introduction to that you've had. And then we're going to talk right at the very end. I'll give you some details about the first lecture quiz, which I believe is scheduled for the 12th of February. So I'll tell you about some of the things that I'm planning to do then. It is, as you may imagine, a work in progress that I have not yet finished. And we begin. We begin. First of all, with congratulations that you may have seen on Twitter that Sherry has started receiving, or your course link, you may have seen that Sherry has started receiving your nasal strips. So well done you. Thank you very much for getting that back. And she's also had the opportunity, as one does over the weekend, to start searching through those nasal strips, and we have found some mites. So if you remember your code, these are some codes where we have already had the opportunity to look through and to see some mites. So you might want to go, if that's your code, you might want to go and take a look at some of the amazing biodiversity that you are host to. A reminder that if you haven't already sent back in your envelope, there's still time. Just do it, please, ASAP. Another reminder from the labs and from Sherry and from that's available on CourseLink is to remember that before you attend your lab this week, make sure to watch the Ripley's Aquarium video and complete the worksheet that's posted under Lab 3 under Content. It sure made Sherry wish that she was diving somewhere warm. Me too, even though I'm not a diver, but somewhere warm would be good. And the lab handout and image document were updated last night, so please give a read of those before you head to your lab this week. All right, let's dive in. So this is where we're going today. This is our last part of a day with the Cnidaria. We're going to begin to explore the Ctenomorphs on Wednesday, which is this clade right at the top. But today we're spending our last time with the Cnidaria. Now the Cnidaria, there's kind of three main lineages. We've talked about them: the jellies, the cnidocytes, and some of the apomorphes, that the nematocysts, these firing barb cells that unify all of the group. They're radially symmetrical, and within them there's three principal lineages. There's the anthozoans, or the corals, that have no medusoid form and are predominated by or characterized by the polypoid life history stage. And then there's a diverse and other group called the medusozoans, and the classes within that group is where we're going to kind of survey today. And then the last group is a group that blows my mind, because up until relatively recently, systematically speaking, say 15 years ago, this is a group of cnidarians that we historically thought were protists. And so now we know that they are extremely reduced and parasitic cnidarians, which is kind of amazing. Those are the mixozoans. So anthozoans, medusozoans, and mixozoans are the principal three lineages. And today, we've talked about the anthozoans already. Today we're going to talk about the other lineages. So This group here, so we've talked about the anthozoans over there on the left. The uncertain placement, of course, represented by the dashed line for the myxozoa, this parasitic group, and then the medusozoans. So a group containing hydrozoans, cubozoans, and scyphozoans. So we're going to dive into scyphozoans, cubozoans, and hydrozoans and myxozoans today. And I can hear you all, if my window is cracked open, I'm sure those of you who are in Guelph, I can hear you saying, hooray, tell me about the scyphozoa. And I would, of course, like to spend lots of time talking about the scyphozoa, but we're going to have very little time. This is kind of a survey that we're going to do, a zoom through the jellies. So the medusozoans, we have here the hydrozoans, the cubozoans, and the scyphozoans. These are a group where they have predominantly a medusoid stage, so a jelly stage. Some groups retain polypoid forms as well. Some groups do not. But they all have a medusoid form at some stage or another. And their mitochondrial DNA is arranged in a linear fashion here, not in a circular form like as we see in the rest of the animal life. There's a lot of, the Cnidaria are home to an enormous number of strange mitochondrial adaptations. Myxozoans have no mitochondria. We see this linear arrangement of the mitochondria here in the medusozoans, and in many cases extra genes and many genes lost. So mitochondrially, if you like to study or think about mitochondrial evolution, the Cnidaria is probably a group for you. So there's a look at those words that I've been talking about in the text on the right-hand side of the slide. The anthozoans are where we have been, represented by the corals, the polypoid forms that lack medusa. The medusozoans primarily represented on the right-hand side here by medusoid forms, motile or mobile forms that can have a polypoid form, as some of the scyphozoans do. And the myxozoans, we don't see them in this figure because they're very strange. We're just going to have to accept that and celebrate their strangeness. So the first group that we're going to talk about over here are the scyphozoans. The scyphozoans is a group of medusozoans that has, as you see, remember the numbers and the dashes across this phylogeny indicate, this is Brusca et al. Mapping for you traits, apomorphies that define a clade or define a group above that point in the tree, or past that point, let me say, in the tree. So here you can see, let's compare and contrast the branch for the scyphozoans with the hydrozoans. Hydrozoans, when we get to them, have a lot of different apomorphies characterizing that lineage, whereas the scyphozoa over here, just one. And that's this process of strobilation. Strobilation, so there's a look at some of the beautiful scyphozoans. If you toured Ripley, probably you've seen some scyphozoans already. If you've dived, maybe you've seen them as well. Strobilation is the production of ephyra, and we'll see that reproduction, the production of the cutest, perhaps, invertebrate around. So characteristics of this group, one of them are these sense organs. Remember, this is prior to this point, when we talked about different lineages, we talked about groups that had no muscles, had no arrangement or nervous system. And in the Cnidaria, we've seen the appearance and the documentation of both muscles and a nervous system. And here is the first time that we started to talk about sense organs. So this Rhopalia. This is on the edge. There's one just poking out of this edge of the pedalia. So that's kind of the edge of the epidermis where the long kind of tendrils begin to form. There are these cup shapes containing Rhopalia. And this is a derivation of the gastroderm, so the stomach layer or the feeding layer, that seems to be an efficient catch-all for sense, that it has it's a statocyst that can be a mechanoreceptor, a photoreceptor, and a chemoreceptor. So this is the first or the most specialized kind of sense organ that we've seen so far, is this Rhopalia. And there it is there. Now the other, what else do we see here? Well, one of them is this corona mussel, because remember, as we talked about movement and we talked about the jellies moving, about the benefit in this lineage that they've derived because a medusoid form can get up and off the surface or the substrate, can swim around in the water column because of the longitudinal and circular muscles laid at right angles, so it can squeeze itself, and as it squeezes across that mesoglia, it ejects water out and essentially acts as a tiny little jet plane, or a jet engine, let's say. There are gastric filaments within these medusoids that you can see here that bear nematocysts and digestive cells. So as it's moving around, it's a highly mobile thing. You see these complex frilly arrangements in the scyphozoans there, where these gastric filaments bear both nematocysts that bump into fish and immobilize them and then begin to digest them right away and then share that with the rest of the body. So those are the frilly dress of that arrangement underneath the bell of the scyphozoans that you saw in the Ripley's tour or in the earlier photographs today. That's what you're looking at there, is the gastric filaments. There's another look at the rhopalium there and the gastric filaments. So strobilation. This is this tower here. So a brief polypoid stage that contains that turns into strobila.
Life reacts to the blooming sea.
Now we're going to take a little bit and have some amazing footage from the first issue, or the first issuing of Blue Planet, and have Attenborough and the BBC tell us about it. If you're attending asynchronously, remember this is going to get flagged by Google, and so I'm going to have to trim this. So the video is down there in the bottom. And so we're going to watch it together here for under a minute, and then you can go and take a look at this yourself. The link is in, you can see it there on the screen, and it's also in the chat from today's. So everybody take a moment who's here synchronously to wave and think about the people who are attending asynchronously. Here we go. Put it in the chat, but I'm not going to agree. I can tell you that right now. That is a pretty darn cute thing. So furrows develop here. I'll move my mouse across the screen. This is not a figure but from your text, but it's one that I like, so you're seeing it here in the lectures. A furrow develops across the leading edge or the feeding edge of the polyp. More furrows develop kind of equidistant apart. And then these ephyra, these tiny baby medusoids, develop sensory and muscular structures. They start to pulsate and they break free. And once they all detach, this small polyp remains. That's kind of amazing. So the scyphozoans, only one kind of unifying. We've seen the rhopalia, but the strobilation is the predominant apomorphy for this group, and I think it solidifies their position as maybe the cutest of the invertebrates that we have seen or will see. Okay, so I'm going to jump over and vérifier le chat pour une seconde, and what do we see? Not disappointed. I like it. Thank you. Awesome. And I will jump back, and we will leave Caramel the cat alone. And we're going to move from the scyphozoan to the cubozoans. So you can see three different apomorphies characterizing the cubozoans. As the name, zoa or zoid meaning animal, cubo meaning square or cube-like, box-like, that's describing one of the apomorphies for the group. So you remember at the start we talked, and in general we talk about the cnidaria being radially symmetrical. This is one tweaking of that apomorphy consistent for the group, where inside of one of the groups we've seen either the loss or the alteration of an apomorphy that describes the total group or the group in whole. So here, instead of being radially symmetrical, we see almost a box or a square-like form, the medusoid form. The rhopalia here is even more advanced, and there are near lenses that appear, and a velarium. And a velarium is not a Harry Potter incantation, but it's an assist in the jet propulsion. So cubozoans are very mobile. If you've been stung or if you've read about the irukandji or other stinging and mobile jellies, they are cubozoans. So let's take a look at those. So very, very small polyps that lack the setae. Remember those gastric filaments that taxonomists use to count them? This is about as interesting a picture as you can find. So a reduced, very reduced polypoid stage. And cubic arranged, and they have four pedalia, or four tentacles arranged at pedalia at the four corners of the bell. So the pedalia there, and the tentacles extending across. So kind of like that, those four, I don't know, biceps or first segment of a leg, if you wish to anthropomorphize or bilaterionamorphize this radially symmetrical jelly. But long feeding tentacles arranged at four corners off of the box-like form of the cubozoan. Now on those pedalia, there are these rhopalia that have an ocelli. So a photoreceptor that, despite the fact that there is a lens that seems to concentrate and direct light, remember, there isn't cephalization here. We don't see the concentration of the nervous system. We see junctions, but not a cephalization, not a real extreme concentration of the nervous system. It's essentially a neural net around the exterior of the and surrounding the entire body. That's about squeezing these muscles back and forth so that they can move through the water column. But here, there's been apparent selection through time, strict selection because of the complexity of this rhopalia, it would seem, that differentiating light from dark and potentially image forming, has been extremely important. And so that lets us presume a couple of things, that we have other lines, that they're predatory, right? And they are, and that their highly mobile life history is dependent upon perception of where it's occurring in the water column, which, if you're a marine biologist, would make you think predictably, or let you think make some predictions about what kinds of food that it would eat and how it might change itself in the water column in response to any diurnal stages. It also might help you make some predictions about where cubozoans are found in the water column, like, for instance, if someone were to ask you a true-false question about say, here's the statement: the greatest diversity of cubozoans are found in the abyssal zone beneath the penetration of light. Now, just by the presence of this apomorphy, you would probably say false to that statement, right? Because if there's all of this infrastructure embedded in this cubozoan that's all about telling where light and dark is, why would they exist? Why would they expand and diversify in the abyssal zone beneath the penetration of light? A foreshadowing there about how true-false statements can be constructed in the kinds of quizzes that we'll write and do together about this course, and the kinds of questions, practice questions, that you can make for yourself to help you practice. We continue. The last of the specific things about is this velarium. Now, the velarium is just basically, it's I've said there parenthetically at the bottom, it's developmentally different from the velum, which we're going to get to in a second, but it's essentially under the bell. So if this is the bell and these are the tentacles, so under the bell shape, there's a constriction that allows the water to be forced out much more rapidly. So instead of being kind of an amorphous shape of so you're looking up towards the surface, and this is the underside of the medusoid, this constriction, this circular constriction, like a gasket, forces the water out through a smaller opening, meaning, of course, that it can go much more quickly. So again, an adaptation that makes us think these are mobile in a photosynthetic zone in the marine system, and predators. Predator, predator, predator. So that's our zooming through cubozoans. And let me jump back over. Stan Plebe, yes, would a box jelly be a cubozoan then? You are exactly right. It is a fitting name. And essentially cubozoan is you saying box jelly in derived Latin. So well done. All right, let's continue this survey to talk about the hydrozoans. So here we are here. So still in the medusozoans, the hydrozoans, many more apomorphies characterizing this group. Now, one of the things that I've bolded here in the synapomorphies of the hydrozoas, it's called there, in that the text underneath the tree, is I wanted you to notice that in this lineage, this is a great example of loss, of evolution, not by the addition of complicated things, but by departing from an ancestral pattern through the reduction, simplification, or loss. And we've got all three of them here. So we've got relocation, gamete-forming tissue, not in the gastro, but in the epidermis. Real simplification and loss of gut mesenteries. Simplification of middle layer to an acellular mesoglia. Loss of a gastrodermal nematocysts. So this is a simplified form. Now, one of the cool things, we're going to talk for the hydrozoans for a little while here about both their apomorphies, but also about their diversity, because this is a group that has, well, for one, I suppose it's one that you could bump into river systems in and around Ontario. That there is a freshwater hydrozoid that you could see here, the hydra, or in Europe if you're around there. And so instead of being strictly marine, you can see them in the freshwater. The other is that, remember, we've talked about the anthozoans being dominated by the polypoid form, the scyphozoans and cubozoans being highly mobile, reduced polypoid stages, strobilation in the scyphozoans, and real almost extinction of the polypoid stage within the cubozoans. In the hydrozoans, we see both stages and just a gorgeous combination of them. We see extreme specialization of polypoid forms in colonial mobile floats, so like jellies, but essentially colonies of polyps doing different things and moving in the water column. So they've taken the simplification that I gave you early on, saying the planuloid forms, the anemone, the corals, they sit and they have to filter feed. The other ones over here in the medusozoans, they're moving around, they're predators in the water column. The hydrozoans have said, hold my beer, Smith, we're going to take the polypoid form and we're going to make it mobile. So they've steampunked the design of the cnidaria. So this is a velum. So I contrasted the velarium with the velum. They're accomplishing the same thing. They're derived developmentally from different processes. Don't worry about that. They're two names that you can add to your Scrabble arsenal. But this velum is essentially the gasket across the opening that is allowing the water to be ejected more. So mobility and more elegant mobility. So this ring of tissue that directs the flow of water out of the bell. And you're seeing it through there in this transparent drawing from the old text, the Rupert Fox and Barnes text. So Valalia, I believe, is the name for this float. But this is essentially you looking at, this is not a medusoid form. This is a colony of polypoids that we have gastrozooids here feeding, gonozooids, reproductive floats mixed in within, and then these dactylozooids that have defense and prey capture. And so anytime you've seen these massive floats, if you've been lucky enough to see them blow into shore, or even luckier enough to be out in a boat and seeing them, these are the different zooids that you would have run into. So polypoids that have not restricted, they've lived colonially, they've diversified, and this diversification of task has allowed them to sit up and up at the surface or near the surface. And so in this particular one species example, this is one that it's gone to the surface and moves around by this float and sail that extend above the water and get blown around. But there's also siphonophores, which is a particular group of hydrozoans where you see they exist within the water column. So you don't have to think about them being restricted to the surface in that Portuguese manowari kind of thing. But you can see what you're looking at here is a display of nematocyst cells, different shapes and forms, and specialization of sexual and asexual reproduction in zooids of different siphonophores. Siphonophores are kind of magical things. There's a look at the sail here, extending above, but they bioluminesce. And so this is a particularly gorgeous, attractive bunch of jellies that we share the planet with. So, I see no specific questions there in the chat. And now we jump, so we've gone scyphozoans, cubozoans, hydrozoans, and now the uncertainty with the mixozoans. So the synapomorphies aren't actually listed. See that box there with the question mark? Brusca and Fox have played a very conservative line here and said we don't actually know which particular group or morphological features characterize this lineage. The figure that's shown underneath the tree, figure 7.28, gives you a bit of the story. Previously considered to be protist, mixozoans are now viewed as highly specialized parasitic cnidarians. You can see the nematocyst at either end. They have a spore wall, but they look essentially like an infecting cell with kind of needles or harpoons projecting out either end. And until 2015, and I'm going to insist that we think of that as relatively recently, and I'm going to do that because I was teaching this course, and so we talked about it because I was really excited about this when this came out in the Proceedings of the National Academy of Science. They had been not just a protozoan, but a phylum of their own. So they weren't even contained within another large group. They were a large group, uniquely considered to be parasitic protozoans. Salut, Jack. So you can see these polar capsules. This is perhaps the similarity here to these polar capsules is one reason that people thought that those similarities laid them out. If you compare and contrast to some of the parasitic protozoans, the apicomplexans, for example, this was one of the apical complex of the apicomplexans and the polar capsule here. Their reduction in body size, reduction in a lot of the body wall. This is an extreme evolution via loss. But you can see genetically when we started to sample them, this is, if you take a look at the colored phylogeny on the right-hand side and take a look at, so red, this box that I've put in here in the red, those are all the sequences for all different diverse lineages, multiple species of mixozoans. Cnidaria is all of the other groups here in blue and orange. Deuterostomes are us, ecdysozoans are things, all sorts of things, but mollusks and insects and arthropods. Lophotrochozoa, we'll talk about that in a second, but you can see, and then there's sponges and ctenophores and choanoflagellates. So you can see genetically, this says not at all. So where would we expect to see the mixozoans sit in this phylogeny, the colored phylogeny here, if they were protozoans? Well, the answer would be outside of the choanoflagellates with the rest of the protozoan non-metazoan life. So this is an extreme simplification of a multicellular animal down to the state that for hundreds of years of science, we thought they were unicellular parasites. Now, I'm not done with the cool stories because they kept happening. A year ago, the same week that we went into lockdown, so maybe you didn't read about it because, you know, there were some things in the news in early March of 2020. This paper came out in the Proceedings of the National Academy as well, and it characterized the fact that one of the, genetically, one of the examples of reduction that we see in the mixozoan is a complete absence of mitochondria. So amazing, okay? And it's certainly, if you're going to use barcodes, DNA barcodes, for example, to identify life, this is going to be a problem here because not only are they lacking or are there strange things going on with the gene, the cytochrome C oxidase 1 gene that's used for animal barcoding, but in fact, there is no mitochondria. Ali. So let's jump back to the chat for a second, see if that has blown anybody's mind. I'm going to presume all your minds are blown and they've just happened very quietly, very polite Canadian mind blowing. Thank you. Mind blown. Thank you. So far, the hydrozoan is the dominant form, the polyp, as opposed to the medusa. So far, the hydrozoa is the dominant form, the polyp, as opposed to the medusa. Yes. So I see. So I'd restate that and say, is the polypoid form the dominant form in the hydrozoa? In the ones that I introduced you to, yes. And if you wish to survey that more broadly, take a look through that section of the text for your own enjoyment, not because I would expect to test you on like remembering. And a spoiler alert or an Easter egg to our discussion at the back, these exams are going to be, they're open book, open internet, so even if I did ask you the specifics, you could look them up. So why would I do that? Let's let some people in. Hello, people coming in through the chat. And I will jump back over and say, we're going to say toodaloo. In the last 20 minutes, we're going to talk about the next couple of assignments. One of them is your species naming assignment, or naming a species assignment. Why are we doing this? Well, from my perspective, there's just under 2 million named species on the planet. And most of those, we've talked about this already, are invertebrates. Cryptic diversity, diversity that remains to be recognized and described and named, there's like really snarky arguments that proceed in the ivory towers of academia about how many of those species remain yet to be discovered or recognized. It's no fewer than 10. It could be in the hundreds. Most of that which remains to be described from metazoans, multicellular animals and plants, are invertebrates. So in a course where we get to talk about the invertebrate, the morphology and evolution of invertebrates and the diversity of invertebrates, and where one of the very first learning outcome that I've ranked there is an appreciation for the diversity, I think it would be, I was thinking about this in preparing for this iteration of the course and thinking this is kind of crazy that we don't have take some time to talk about the process of naming, since most of the jobs of naming that will remain in my lifetime and longer in yours and beyond you is that most of the names that remain to be given will be to invertebrates. So to help that, what I'd like to do is to briefly explore some of the why, some of the how, and give you an opportunity to explore the literature of species description to get a feel for the fact that it will appear to be a fairly dry process, but it's in fact super creative. It just has rules, creative within rules. Now, first of all, what I'd like to do this is to have you tell me and us about why it is, why go through the process of naming species. I have some ideas, and you can have no doubt that I will share my ideas about this because I feel kind of passionately about it. But what I'd like you to do is go to menti.com. The code is 581565, and for that to work, I have to go over and open the code and have you tell me about why it is that we would want to see species receive names. I'll try and jump back and 581565. So I haven't put it in the chat. I see lots of you logging in. I see more of you logging in. I'll keep saying it because it interrupts me presenting it, unfortunately, which I think will interrupt you and put a break in how you can start filling stuff in. So I'm reading it. That's what I'm looking down at my lower monitor, and this isn't rude. I'm still making eye contact. And I'll put this up in a second once I see some of the examples of suggestions. And some of them are making me smile. Awesome. Awesome, awesome, awesome. Okay. I think you should still be able to, yeah, you're still being able to fill in there. That's good. So what I'm going to try and do now is change the share so that we can see who's doing what. So let's take a look at, and tell me in the chat, I think you should be able to see the Menti right now. Somebody tell me in the chat, just say, sure, Smith, we can see the Menti. We can see the Menti, we can see, thank you. All right, so let's take a look at these. For easier communication between scientists, for conservation purposes, a derived or a specific example of the second, to differentiate, to identify, to categorize, to make communication about species more universal, to make sure everyone is on the same page and talking about the same thing, to keep track of all the different kinds, so we can truly understand species in the world and help maintain a sense of order. So kind of an anthropomorphic reason there. Helps to discuss specific species with less confusion, gives an insight into what they are related to. It's fun. I agree. Gives insight as to what they do. Unique locomotion, shape, organize, species. Now, I'm going to take some screen grabs of this, and they're going to be included in the PowerPoint, the PDF that eventually gets up. But all of these things, trace evolutionary lineages and make it easier to identify them in person because it's fun. And there were a couple things in here that talked about the utility of doing it, but also then about maybe a specific need that we have to categorize things. And I think I like all of these examples. And let's go through and talk about, so as I'm doing this, put your hands up in the air, maybe even stand up, give a big stretch, and pat yourself on the back, because I like basically all of the things that you came up with. Okay, so there I've tried to go back. Can you tell me in the chat whether you can see the PowerPoint slide again in kind of a nice peach? Yep, thank you. Okay. So I like all of those. I think in a nutshell, naming allows us, so putting aside whether or not we feel a need or whether there's been an evolutionary pressure in our own history that rewards those of us that can identify things that are good and bad, that hurt us or help us, because I think that's a good anthropogenic kind of a lineage or a discussion. But naming allows us to understand, document, and share knowledge. It's impossible for us to proceed scientifically as a way of asking questions and understanding the world unless we have a shared lexicon of what we're talking about. And this is why I put the image of the Tower of Babel in the bottom, that we want to make sure that we're talking about the same thing. And so in creating a name, a scientist can tell a story about the species and about other things in their life that have been important to them, about a person or a place being honored. It lets the scientist kind of tell a story about themselves. And so Steve Hurd, a colleague and friend who wrote an amazing book about naming species, talked about the fact that this invention of Linnaeus essentially allowed a window into scientific personalities. I'm going to talk briefly about these things. So, some of the rules involved in naming species. There is actually a code. This is the International Code of Zoological Nomenclature. Not only is there one code, there's multiple, because fungal people have theirs, plant people have theirs. This is for animals. I'm only going to talk about this one. And this is if you're looking to fall asleep at night, you could go and read this code. But a species name is essentially a combination, and you know this, but these rules are actually written out. So if you're going to write a new one, you have to know the rules. It's a combination of two names, or a binomen, or a binomial, a scientific binomial. Perhaps you've heard it described. The first is the genus, or the generic name, and the second is the specific. And we write them, the genus has to be referred to in the uppercase. The species always begins with the lowercase, and they're italicized when they appear together, or just the generic name on its own. Why are they italicized? That actually has nothing to do with science. It has to do with printmaking and the fact that historically, whenever a secondary language was used in print, that it was offset from the principal language in question visually by italicizing. And so Latin names and scientific names, which you may have heard called Latin names, are Latinizing or Greekizing words or places, and so they would not be English. And so if English was the normal language, or French or German or whatever, and so they're always been italicized. there are specific rules in this 400 to 700 page version of the code, and one of them is that the authors have responsibility. They're not allowed, it's strongly suggested that they don't do the distasteful and selfish thing of naming something after themselves, although that isn't actually a rule. It's just a best practice that's followed. And that authors should exercise care and consideration in forming new names to ensure they are chosen with subsequent users in mind, and as far as possible are appropriate, compact, euphonious, memorable, and do not cause offense. It's kind of the same list of things that you would think about when you're naming your cat or your puppy or your child. So appropriate, compact, you want to be able to say, well, you don't want to name your cat Jellyfish, perhaps. And compact, you don't want to name it a long name that's hard to call if the cat has escaped outside. Euphonious means it sounds good. Memorable means you can remember what you called your cat, and you don't also want to call your cat, you could call your cat WTF, but you wouldn't want to split off the last F and call your cat that, because calling it in the neighborhood might give offense. Ta-da. So, many rules with much room for creativity. So in the assignment that's going to be released, what I'd asked you to do is to find examples, and to tell me about the etymology. What was the species named for? That's actually a section in a species description. So when you find one online, you can Control F for the word etymology. It will then have a section to say the specific of this binomen is in honor of my dog. Da-da-da-da. The distribution is important. Was it just found in one place? There's actually some interesting statistics about most invertebrates species being described, or an increasingly large proportion compared to vertebrates, that are described from only one place, and in some cases from only one sample. Now think about that. If it's only one sample, how do we differentiate males and females if they're split like this? The deposition means that type specimen, which is the classic, the one that we're going to agree to refer to when we use this name in the future, has to be put somewhere public, in a collection, in a museum. So it has to be checkable. Paratypes means that there's more there in the type series, in this description. So we have a type specimen, a holotype, the classic, the archetype of what this species is. And we have maybe 7 others that came from the same place, and we're going to include them in this description to try and capture some of the intraspecific variability that's inherent to all species. And then how did we differentiate it? Different papers and different descriptions have different ways of doing that. Sometimes it's just with DNA. Sometimes it's with DNA and photographs and measurements. Sometimes it's actually with a key. So you remember when we were talking about the Placozoans, the phylum formerly of 1, in the paper that I presented then that was published in PLOS Biology 2 years ago, this is actually the description where Hoylungia hongkongensis was described. And you can see here that it has a type locality, that it was isolated from the Chung River close to the small mangrove in this particular village inside of Hong Kong, and a particular date. Excuse me, more than that, in this case they've even given a grid reference that you can go, if you wish to resample this new species, that's exactly where it was found. The type specimen was deposited in this large German name that I won't go into, in collection in Germany. And the etymology, Hongkongensis, means from Hong Kong. I'm excited enough, I'm going to cough for a second. So this is one case where the name is an honorific of where it was found. You can have others. This is a species of snail that was described about a decade ago in the journal Zootaxa. And it's an honorific in both parts of the binomen, the generic and the specific. This is Crikey steve-irwini, which is a new genus and a new species from the montane wet tropics of northeastern Queensland, describing or honoring an expression of Steve Irwin and Steve Irwin himself. Now, as it happens, I'm part of an authorship group that has published a species description today for 400 new species of parasitoid wasp from Costa Rica. I apologize for the coughing. And this is one example. So one of those 403 species, the description is all here. This is a very brief and kind of a reductionist form of species description, where we've done kind of the minimum of what it needs to do to describe a species. Jack, ça c'est superbe. J'aime ça. J-A-C-K, Jack.
Oh, so you could see something about all of the letters.
Yeah. Okay.
That I colored it in and I made shapes in it.
Super good. Tu vas le montrer, maman? Super good. So, Harry Ramirez is a parataxonomist, a colleague and a friend who works, a Costa Rican who works in the protected area where I work in northwestern Costa Rica. And this is Harry and I standing beside a Malays trap in the Bosque Arenales, which is a mature rainforest on the Pacific shore of northwestern Costa Rica, within rough spitting distance of Nicaragua. The diagnosis here for this particular species, merci monsieur, is two forms only, DNA and this picture. And that's partially because parasitoids, which is we'll talk about when we get to insects, are a group of insects that develop and lay their eggs inside of or on top of a species like a caterpillar, a butterfly or moth, develops within it, and then they make a tiny black wasp that has virtually no morphological discontinuities upon which you can hang a name. So much of the vast diversity of life that remains to be described are parasitoids. And so in describing many of them in this reductionist form, we're trying to accelerate the rate with which we describe these. So we've used DNA and a photograph. Now we've also included other things. So the holotype here, so the type specimen, which that picture is of, came from a particular section of the Rincon de la Vieja, so a particular volcano that's about 20 kilometers east of where we're standing right there. We have host data about what it emerged from. We have a link to information about that on a collection website. We've seen, and then there's the etymology. So Chilonis Harry Ramirez E is named to honor Senor Harry Ramirez of Guanacaste Dry Forest Conservation Fund in the Area de Conservación Guanacaste for his many decades as a dedicated inventory parataxonomist, Malays, and mouse live trapper and species processor within the ACG. So it's honoring the work that he's done in documenting and preserving the biodiversity of this part of Guanacaste. So that's the etymology here. There's the description about the holotype. It's deposited actually in Canada in the Canadian National Collection of Insects, Arachnids, and Nematodes, which is in Ottawa on the Essential Experimental Farm. There's the diagnostic of a particular stretch of mitochondrial DNA, the DNA barcode region. And there's the photograph. And we've done this a lot in my lab and with collaborators around the world. Other particular example, we named a bunch of species of these wasps after school children in Guanacaste, school children who had won a contest where they had to write an essay or draw a picture about wild nature and about how wild nature affected them and how they interacted with it. So this is I love this picture, and you can read more about it, and I'll put up some links to some popular press articles that were written about this process as we named species of wasp in these forests after the people who live in and around the forests in this national park. And because 1.9 million names, maybe 100, 200 to go, even the dregs or different people amongst this, you can all have species named after you. This is actually 5 different ones that are named after me. Hooray! Woe to the wasp. Now, what are we asking you to do? We're going to ask you to search Google Scholar for 1 or 2 open access journals, so you don't have to be on a VPN to the university to do this. One of them is called PeerJ, one of them is called Zookeys, that regularly publish descriptions, and then follow these steps. Okay, you're going to have these steps in a second. I'm going to show them to you. So those links, and you're going to have these links in the assignment. You click on them, and it will open a page like this. So here's the one where I've searched in Google Scholar, new species from Zookeys. As a hint, this is also how you use Google Scholar to search specifically within one source. You can make it even more specific, which is one of the things we're asking you to do in this assignment, by adding plus and a taxa name. This is a type of beetle, a family of rove beetle. And if I click on this very first example there by A.J. Brunke, it takes me to the fact that A.J. Brunk, to this particular paper that Adam Brunke published in Zookeys in 2012, where he described new species of staphylinid from Canada and from Ontario, and including from the Guelph Arboretum. And Adam is now a taxonomist at the Canadian National Collection, and you're going to meet with him during your labs at some point this year. So, amazing. Jack is definitely killing it. Jack is doing pretty well at school. 1 minute left. So, very briefly, I want to tell you a little bit. We've got time. This is still like this is the first today. This is the exam that's on the 12th. I just want to give you some details about the lecture exam. You all have 2 hours to write it. You can start any time on Friday the 12th after 9:30 a.m. It's designed historically, if we were together face to face, you'd be writing it in the exam time. So you all have 2 hours to write it once you start. That starts the timer. But I have written an exam that is designed to be done in 40 to 50 minutes. So you probably don't need it. So anyone who's got SAS considerations, consider them all accepted. You all have double time to write this if you want it. But life's too busy. If you don't need it, please do not take 2 hours to write it unless you need it. The exam has to be done by 5:30 p.m. So you have the working day on February 12th to do it. You have 2 hours, but you shouldn't need it. It's going to be on CourseLink, in the quiz format on CourseLink, and it's going to be comprised of true and false questions. It's going to use material from the lectures, so focus on the lectures, use the textbook secondarily. I'm then going to go out into the interpipes and pick up figures and maps and diagrams or phylogenies that I'm going to include. And I'm going to ask you questions about concepts and examples and things that we've talked about in the lecture to date. But what I'm trying to do is to write an exam that's kind of uncheatable, because it is open book, it's open internet. So if you're confused by some element of the question, please Google it. There's no respondus, because that's a violation of your privacy, and it's a violation of trying to do good things. And the lecture periods will cover up to the end of the day Friday. And what I'll do on Wednesday is I'll put a couple of examples up, and we'll do them together in lecture on Wednesday. So with that, I want to say apologize, because I think I've gone a minute over, and you've got places to go and things to do. So I encourage you to get outside and read something for pleasure, watch something for pleasure, do something for fun and for exercise, get up and move around, take care of yourself, take care of those of you that you are with, and we will see you on Wednesday. And some of you will have had your lab by then, so enjoy that. All right, talk to you soon. Bye-bye. I am going to hang out for a second and keep answering questions in the chat if you're interested in that, or seeing if I haven't caught some of your questions yet. So take care.